Inside the quiet darkness of your skull lies the human brain, the most complex object in the known universe. It functions as a biological supercomputer that never truly powers down. This 3-pound mass of tissue consumes 20% of your body's total energy despite accounting for only 2% of its weight. If you find these insights helpful for your bedtime listening, feel free to subscribe for more science for sleep. The organ manages every heartbeat and memory while remaining entirely incapable of feeling pain itself. Because the brain lacks nociceptors, surgeons can perform complex procedures on a conscious patient without them feeling a single touch on the gray matter. It is the seat of all sensation, yet it exists in a sensory vacuum of its own making. Every flicker of light you see and every sound you hear is merely an electrical interpretation generated within this silent chamber. To understand its power, we must look closer than the naked eye allows. The architecture of the brain is built upon roughly 86 billion neurons, each one a tiny messenger firing pulses of electricity. These cells form a web so dense that a single cubic millimeter of cortex contains nearly 1 billion synapses. If we were to unspool every nerve fiber in a single adult brain, the resulting line would stretch over 100,000 miles. Such a distance is enough to circle the Earth 4 times, all tucked neatly inside a space no larger than 2 clenched fists. These pathways are not static. They are constantly rewiring themselves in a process called neuroplasticity. Every new thought or skill creates a physical change in the cellular landscape. We are essentially walking monuments to our own experiences, sculpted by the very signals we process every second. The internal universe operates on a scale so minute that thousands of its components could fit on the head of a pin. Within this microscopic realm, the speed of information transfer reaches nearly 270 miles per hour. Imagine 86 billion individual cells, each one a specialized processor working in a massive, coordinated grid. These neurons form the foundation of every memory you hold and every dream you experience during bedtime listening. Each neuron possesses a main body containing its genetic code, but its true power lies in its extensions. The dendrites reach out like the intricate branches of a winter oak, catching incoming signals from thousands of neighbors simultaneously. On the opposite end, the axon serves as a biological cable, carrying an electrical impulse away from the cell center to distant targets. Have you ever wondered if your own thoughts feel more like a spark of light or a flowing river? This architecture is surprisingly vast, with some axons traveling the entire length of your spinal cord despite being microscopic in diameter. To protect these signals, a fatty substance called myelin wraps around the axon like insulation on a copper wire, ensuring the message does not degrade as it travels. When the electrical pulse reaches the end of its journey, it encounters a tiny physical chasm known as the synapse. At this junction, the process transforms from electrical to chemical as neurotransmitters flood the gap to dock with the next cell. This elegant handoff happens trillions of times every second, creating a biological internet more complex than any man-made machine. If we were to unravel every nerve fiber in a single adult brain and lay them end to end, the resulting thread would stretch for roughly 100,000 miles. That distance is enough to circle the entire planet 4 times over, tucked neatly inside the protective curvature of your skull. This dense packing is made possible by the extreme thinness of the fibers, which are often measured in microns. While neurons get most of the credit, They are supported by glial cells that act as a maintenance crew, cleaning up waste and providing structural stability. The sheer scale of this connectivity is difficult to comprehend, as a single cubic millimeter of cerebral cortex contains nearly 1 billion synapses. Some estimates suggest there are more connections in your head than there are stars in the Milky Way galaxy, a fact that bridges the gap between the microscopic and the cosmic. Every decision, from the twitch of a finger to the deep processing of science for sleep, relies on this electrochemical harmony. It is a system that never truly shuts down, even as you rest, constantly pruning and strengthening its pathways to reflect your life's experiences. The energy required to power this network is surprisingly modest, operating on about 20 watts, barely enough to light a dim bulb, yet it manages the most complex data processing known to exist. This efficiency is a hallmark of biological engineering, allowing for massive parallel processing without overheating. Within this network, specific clusters of neurons are dedicated to specialized tasks, yet they remain in constant communication with distant regions. Such interconnectivity ensures that a single scent or sound can trigger a cascade of related memories across the entire structure. While neurons often capture the spotlight in our understanding of the mind, they are actually outnumbered by a silent, complex scaffolding of support cells known as glia. For decades, the scientific community largely viewed these cells as mere glue that held the neural architecture in place. But modern research published in journals like Nature Neuroscience reveals a far more active role. These cells are the unsung architects of every thought and movement, performing the vital maintenance that allows the brain to function as a coherent whole. Imagine a vast city where neurons are the residents, but glial cells represent the entire infrastructure: the power lines, the waste management, and the security forces. Star-shaped astrocytes are perhaps the most versatile members of this hidden majority. They anchor neurons to their blood supply, carefully regulating the flow of oxygen and nutrients to ensure the brain remains fueled without being overwhelmed. By maintaining a precise chemical balance in the extracellular space, astrocytes prevent the toxic buildup of neurotransmitters like glutamate, which could otherwise lead to neural exhaustion. They act as the brain's internal pharmacists, dispensing the exact ions and molecules required for a single spark of thought to travel clearly. Then there are the oligodendrocytes, the master insulators of the central nervous system. These cells wrap their lipid-rich membranes around the long axons of neurons to create the myelin sheath, a biological miracle that increases the speed of electrical signals by up to 100-fold. Without this specialized insulation, our reactions would be incredibly sluggish, and the high-speed processing required for language or motor skills would be impossible. It is fascinating to consider how much of our cognitive intelligence relies not just on the cells themselves, but on the quality of this microscopic wrapping. Do you ever find yourself wondering if your most vivid childhood memories are the ones with the most durable neural insulation? Defense within this dense landscape is handled by the microglia, the brain's resident immune specialists. They are constantly on the move, scavenging for damaged neurons, plaques, and potential pathogens that could disrupt the delicate internal environment. Unlike the immune cells found elsewhere in the body, microglia are uniquely adapted to navigate the brain's intricate forest without causing unnecessary collateral damage. During our early years, they even serve as gardeners, pruning away weak or redundant synaptic connections to help sculpt a more efficient mind. This invisible workforce ensures that the brain's high energy demands are met with even higher standards of efficiency and protection. By managing the infrastructure of consciousness, glial cells transform a collection of individual neurons into a unified, high-performance supercomputer. The speed at which these internal messages travel is not uniform, as the brain prioritizes certain types of information over others to ensure survival. This selective acceleration is made possible by the myelin sheath, a fatty white substance that wraps around axons like high-grade insulation on a copper wire. Without this coating, electrical impulses would leak and dissipate, crawling along at a sluggish pace. Instead, myelin allows signals to perform what neuroscientists call saltatory conduction, where the charge literally leaps between gaps in the sheath known as the nodes of Ranvier. These jumps can propel a signal at speeds exceeding 250 miles per hour, roughly the same velocity as a high-speed bullet train. Have you ever noticed how quickly you can catch a falling object before you've even fully realized it was slipping? This near-instantaneous awareness stems from our proprioceptive fibers, which are heavily myelinated to provide constant updates on the body's position in space. In contrast, the pathways for dull, aching pain, known as C fibers, are often unmyelinated, meaning that a stub toe might take a full second or two to register its true intensity in the conscious mind. While many signals must reach the cerebral cortex for interpretation, the most critical survival responses never even wait for the brain's permission. This phenomenon is known as the reflex arc, a neural bypass where sensory information enters the spinal cord and immediately triggers a motor response through a local circuit. When you touch a hot stove, your hand recoils before the heat signal has finished its journey to your head, a protective lag reduction strategy that saves precious milliseconds of tissue damage. There is a distinct difference between this mechanical reaction time and cognitive processing speed, which involves the complex integration of multiple sensory inputs. While a reflex can occur in under 50 milliseconds, a conscious decision, like choosing which word to say next, requires a much more extensive round trip through the brain's associative centers. Even within the brain itself, the white matter tracts serve as the essential biological fiber optics, ensuring that the left and right hemispheres remain in constant, lightning-fast communication. This vast network of biological wiring ensures that our internal world remains synchronized, even when the external world moves with unpredictable and dangerous speed. This efficiency creates a seamless experience of reality, where the delay between an event and our perception of it is practically imperceptible. The biological machinery driving this perception is notoriously demanding, operating as the most metabolically expensive organ in the human body. While it accounts for roughly 2% of your total body mass, this 3-pound cluster of tissue consumes a staggering 20% of your daily caloric intake. This means that 1 out of every 5 bites of food you eat is dedicated solely to fueling the electrical storms occurring behind your eyes. If we translate this chemical energy into electrical terms, the brain runs on approximately 12 to 25 watts of power. It is a humbling thought to realize that the most complex structure in the known universe operates on roughly the same amount of electricity required to power a dim LED bulb in a hallway. Have you ever felt that distinct mental fog after a long day of deep concentration, perhaps wondering if your brain has simply run out of fuel? Even during periods of intense intellectual labor, the total energy consumption of the brain doesn't actually spike as much as one might expect. Instead, the organ reshuffles its resources, diverting power to specific localized regions while dimming the activity in others. When the body faces extreme caloric deficits or starvation, the brain asserts a strict hierarchy of survival. It remains the last organ to lose weight, as the body will systematically break down muscle tissue and even bone to ensure the neurons receive a steady supply of glucose. This prioritization is a testament to its status as the executive command center, where even a brief interruption in energy flow can lead to irreversible cellular damage. To maintain this high-performance output without self-destructing, the brain relies on a sophisticated internal radiator system. Because electrical activity generates heat, the organ would quickly cook itself if not for the constant circulation of blood and cerebrospinal fluid. This fluid bath serves a dual purpose, acting as both a nutrient delivery system and a thermal regulator that carries excess heat away from the dense neural clusters toward the surface of the skull. Scientific observations using thermal imaging suggest that the brain's temperature can fluctuate significantly based on circadian rhythms and sleep cycles, yet it remains within a very narrow, safe margin to prevent protein degradation. This constant thermal management is what allows you to sustain deep focus for hours without the delicate biological components warping under the pressure of their own metabolic fire. Even as you rest, the brain continues this high-wire act of resource allocation, proving that its true power lies not just in its processing speed, but in its ability to manage a limited energy budget with unrivaled precision. The architecture of this 3-pound universe is divided into specialized regions known as lobes, each possessing a distinct set of responsibilities. The frontal lobe, situated directly behind the forehead, acts as the brain's executive suite. It is responsible for the high-level decision-making, planning, and personality traits that define a human being. Deep within this region lies the motor cortex, a thin strip of tissue that generates the electrical signals required to move every muscle in the body. When you think about reaching for a glass of water, the frontal lobe is the conductor initiating the symphony of movement. Just behind the frontal lobe sits the parietal lobe, which serves as the primary hub for sensory integration. It processes information regarding touch, temperature, and pain, while also calculating spatial relationships. This allows you to navigate a dark room without bumping into furniture, as the parietal lobe maintains a constant internal map of your physical surroundings. To the sides, near the temples, the temporal lobes function as the archives for memory and the processing center for sound. This area houses the hippocampus, a structure vital for turning short-term experiences into long-term memories. At the very back of the skull, the occipital lobe is dedicated almost entirely to vision. It receives raw data from the eyes and translates it into the shapes, colors, and motion that form your perception of reality. These lobes are arranged across 2 hemispheres that look like mirror images but operate with subtle differences. Connecting these 2 halves is the corpus callosum, a massive bridge consisting of over 200 million nerve fibers. This structure facilitates a constant, lightning-fast exchange of data, ensuring the left and right sides of the brain act as a unified whole. You may have heard the popular idea that people are either logical left-brained thinkers or creative right-brained artists. Neuroscience has largely debunked this as a myth, revealing that almost every complex human activity requires both hemispheres to work in harmony. While some functions like language are often centered in the left hemisphere, The right hemisphere is simultaneously processing the tone and emotional context of those words. Do you ever find yourself noticing the rhythm of someone's voice more than the actual words they are saying? To safeguard these delicate structures, the brain is encased in 3 protective layers called the meninges. The outermost layer, the dura mater, is a tough, leather-like membrane that provides a sturdy barrier against injury. Beneath it lies the arachnoid mater, a web-like structure that creates a space for cerebrospinal fluid to circulate. The innermost layer is the pia mater, a thin, delicate veil that clings closely to every curve and fold of the brain's surface. This entire system is bathed in cerebrospinal fluid, which serves a dual purpose. It provides buoyancy, allowing the brain to float within the skull so that its own weight doesn't compress the blood vessels at the base. It also acts as a hydraulic shock absorber, cushioning the neural tissue from the impact of sudden movements. This fluid is constantly being produced and reabsorbed, creating a slow, rhythmic tide that clears out metabolic waste products and maintains a stable chemical environment. Every fold and groove on the surface, known as gyri and sulci, exists to increase the surface area of the cerebral cortex, packing more computational power into the tight constraints of the human skull. This massive expansion of the cortical surface is nowhere more evident than in the primary visual cortex, located at the very back of the head. It might seem counterintuitive that the machinery for sight is situated as far from the eyes as possible, yet this arrangement allows for a sophisticated journey of data through the brain's interior. Nearly 30% of the human cortex is dedicated to processing visual information, a staggering allocation of biological resources compared to the mere 8% used for touch and the 3% used for hearing. This dominance reflects the sheer computational demand of translating a 2-dimensional mosaic of light into a 3-dimensional world. When photons enter the eye, they strike the retina, a thin layer of neural tissue that serves as the biological equivalent of a camera sensor. Here, approximately 126 million photoreceptors engage in a process called transduction, converting the physical energy of light into a language of electrical impulses. These signals travel along the optic nerve, passing through the lateral geniculate nucleus, a relay station in the thalamus, before reaching the occipital lobe. The brain does not simply project an image onto the back of the skull like a cinema screen. Instead, it deconstructs the world into fundamental components. In the early stages of visual processing, specific clusters of neurons are tuned to detect very particular features, such as the orientation of a line or the contrast of an edge. Scientists like David Hubel and Torsten Wiesel discovered that some cells only fire when they perceive a vertical bar, while others react exclusively to horizontal or diagonal lines. As these basic inputs move forward through the visual hierarchy, they are recombined into increasingly complex shapes and patterns. This modularity allows the brain to process different qualities of an object simultaneously and independently. One specialized pathway, often called the ventral stream, handles the what of an image, identifying colors and shapes to recognize a face or a familiar tool. Meanwhile, the dorsal stream focuses on the where, calculating motion and spatial location. This parallel processing is so distinct that a person with specific brain damage might be able to see an object clearly but lose the ability to perceive its movement, a rare condition known as akinetopsia. In the quiet moments of bedtime listening, you might consider how your brain maintains such a stable view of reality despite the constant movement of your eyes. Every time your gaze shifts, the image on your retina jumps violently, yet your perception remains steady and fluid. This is due to a predictive mechanism called a saccadic suppression, where the brain briefly shuts down visual input during eye movements and fills in the gaps with its best guess of the surroundings. This internal simulation extends to the physiological blind spot. There is a point in each eye where the optic nerve exits the retina to head toward the brain, creating a small area devoid of light-sensing cells. Under normal circumstances, you never perceive a hole in your vision because the brain performs a feat of spatial interpolation. It looks at the texture, color, and patterns surrounding the void and seamlessly paints in the missing data. This hidden superpower ensures that your conscious experience of the world feels complete and uninterrupted, even when the raw sensory input is fundamentally flawed. These relaxing facts remind us that what we see is not a direct transcript of reality, but a highly curated and polished masterpiece of neural engineering designed to help us navigate the world with effortless grace. Through this lens, the act of simply opening your eyes becomes one of the most complex feats of physics and biology ever performed in the known universe. A constant stream of science for sleep that continues even as you begin to drift off. While your eyes parse the light, your ears are performing a parallel miracle of mechanical translation. Deep within the inner ear, the spiral-shaped cochlea houses thousands of microscopic hair cells that act as biological sensors. When sound waves ripple through the fluid-filled chamber, these delicate structures bend, opening tiny ion channels that convert mechanical vibrations into the electrical language of neurons. It is a precise arrangement where different areas of the cochlea respond to specific frequencies, creating a tonotopic mapping of sound that travels toward the brain's auditory cortex. This process is so sensitive that the movement of these hair cells can be measured on a scale smaller than the width of an atom, allowing for the detection of the faint rustle of leaves or the low hum of a distant engine. This raw data stream enters a landscape of chaotic noise, yet the brain possesses a superpower known as auditory filtration. You might have experienced this in a crowded room, where dozens of voices compete for your attention, yet you can lock onto a single conversation with startling clarity. This phenomenon, often called the cocktail party effect, relies on the brain's ability to selectively suppress irrelevant frequencies while amplifying the specific harmonic patterns of a chosen speaker. The auditory cortex works in tandem with the thalamus to act as a gatekeeper, deciding which vibrations deserve your conscious awareness and which should fade into the background. Have you ever noticed how your own name seems to pierce through even the loudest ambient noise? This suggests that even when we aren't consciously listening, our brain is constantly scanning the environment for high-priority information, a vital survival mechanism that keeps us tethered to our surroundings. Beyond just identifying what a sound is, the brain must determine where it originates in three-dimensional space. To achieve this, it employs two primary calculation methods: interaural time differences and interaural level differences. If a sound comes from your left, it reaches your left ear a fraction of a millisecond before it reaches the right. The brain's superior olivary complex detects this infinitesimal gap, which can be as small as 10 microseconds, allowing you to pinpoint a sound source with incredible accuracy. Simultaneously, the head acts as a physical barrier that slightly muffles the sound reaching the far ear, providing a volume cue that further refines this mental map. This spatial processing is so ingrained that we can often visualize the layout of a room just by the way echoes bounce off the walls, a skill known as echolocation that exists in varying degrees in all humans. This mastery of sound is inextricably linked to our sense of physical equilibrium. The inner ear is not just for listening. It houses the vestibular system, a complex network of fluid-filled loops called semicircular canals. These canals detect the rotation of your head, while other structures called otolith organs use tiny calcium crystals to sense gravity and linear movement. Because the auditory and vestibular systems share the same neural pathways, our sense of where we are and what we hear are deeply intertwined. This synergy ensures that as you move through your environment, your brain maintains a stable horizon while simultaneously tracking the shifting landscape of sound around you, creating a seamless experience of being present in the world. The sense of smell operates through a biological architecture unlike any other sensory system in the human body. While your eyes and ears must send their data through the thalamus, which acts as a grand central relay station, odors bypass this gatekeeper entirely. This direct pathway allows chemical signals from the air to reach the brain's primary processing centers with incredible speed and raw, unfiltered intensity. When you inhale, volatile molecules bind to specialized receptors in the olfactory epithelium, a small patch of tissue high inside the nasal cavity. These receptors send electrical impulses straight to the olfactory bulb, which sits just beneath the frontal lobe. From this vantage point, the information travels directly into the limbic system, an ancient and fundamental part of the brain responsible for our deepest emotions and survival instincts. The olfactory bulb shares an intimate physical border with the amygdala, the almond-shaped structure that processes fear, pleasure, and emotional significance. It sits just as close to the hippocampus, the critical engine for forming new memories and retrieving old ones. This architectural proximity explains why a sudden whiff of cedar or a particular perfume can instantly transport you to a specific afternoon from decades ago. These occurrences, often called Proustian memories, are typically more emotionally charged and vivid than memories triggered by sight or sound alone. Evolutionary biologists suggest this immediate link developed as a vital survival mechanism, allowing early humans to instantly recognize the scent of a predator, a fire, or toxic food without waiting for the slower, higher-order cortical processing to catch up. The genetic blueprint for this system is staggering in its complexity and scale. Humans possess roughly 400 different types of functional scent receptors, each finely tuned to detect specific molecular shapes and chemical functional groups. Remarkably, about 3% of the entire human genome is dedicated solely to the instructions for building these olfactory receptors, highlighting how central this sense has been to our species' development. This genetic investment allows the brain to distinguish between an estimated 1 trillion different odors, a range of detection that far exceeds our ability to differentiate between colors or musical tones. Each individual carries a slightly different set of these active genes, meaning the way you perceive the scent of a morning forest or a rain-slicked sidewalk is genetically unique to you. Even the neurons in this system are extraordinary. They are among the few in the entire nervous system that regularly regenerate throughout your life, ensuring that your chemical connection to the world remains sharp. This ancient chemical sense provides a constant, invisible map of your environment, anchoring your life story in the physical reality of the molecules that surround you. Beyond the molecules you breathe and taste lies a silent, internal awareness that functions as your true sixth sense. This is proprioception, the sophisticated neurological system that informs your brain of your body's position and movement without requiring a single glance from your eyes. It is the reason you can reach out and touch the tip of your nose in a pitch-black room with perfect accuracy. This internal mapping relies on a hidden network of specialized sensors called proprioceptors, which are embedded deep within your muscles, tendons, and joint capsules. Think of these as biological tension meters, constantly feeding data to your central nervous system about the stretch and contraction of every fiber. Muscle spindles monitor the length of your muscles, while Golgi tendon organs track the force of every movement, ensuring your limbs do not overextend or snap under pressure. Your brain integrates these mechanical signals with information from your inner ear and even the stretching of your skin to create a real-time, three-dimensional model of your physical self. This subconscious body schema is what allows you to climb stairs while looking at a friend or type on a keyboard without watching your fingers. Have you ever noticed how you can feel the exact end of a pen or a tool as if it were an extension of your own hand? This phenomenon occurs because the brain is so adaptable that it can actually incorporate external objects into its internal map, expanding your sense of self beyond your physical skin. The cerebellum serves as the primary processing hub for this data, executing millions of calculations per second to maintain your balance and posture without you ever having to think about it. However, the true power of proprioception is most visible when it is absent. In rare cases of neurological damage or specific vitamin deficiencies, individuals may lose this internal feedback loop, leading to a sensation of being a disembodied mind. Without proprioception, a person might look at their own arm and feel as though it belongs to a stranger, unable to move it unless they are staring directly at the limb to guide it through sheer visual willpower. It highlights a fundamental truth about human existence. Your brain does not just live inside your skull. It is woven into the very fabric of your muscles and joints. This silent dialogue between your brain and your body is what gives you the grace to move through the world, providing a foundational sense of presence that persists even when the lights go out. It is the anchor of your physical identity, a masterpiece of coordination that operates entirely below the level of conscious thought. Deep within the parietal lobe lies a specialized strip of tissue known as the somatosensory cortex, a command center where the brain maintains a meticulous, topographic blueprint of your entire physical form. This neural rendering is often referred to as the cortical homunculus, a term meaning little man, which describes the way your brain perceives the geography of your skin and limbs. If you could see yourself through the eyes of this map, the proportions would be unrecognizable. You would appear as a creature with enormous, sweeping hands and massive, sensitive lips, while your torso and legs would shrink into thin, spindly afterthoughts. This distorted image exists because the brain does not allocate space based on the size of a body part, but rather on the density of the sensory information it provides. Your fingertips are packed with thousands of specialized mechanoreceptors that allow you to distinguish the microscopic difference between velvet and satin, and such high-resolution data requires a massive amount of dedicated processing power. The lips and tongue are similarly prioritized, demanding vast neural real estate to manage the intricate mechanics of speech and the vital task of evaluating food. In contrast, the skin on your back is relatively sparse in its sensory hardware, meaning a large area of your trunk might be represented by only a tiny, narrow sliver of brain tissue. This internal map is far from a permanent engraving. It is a dynamic, shifting landscape that responds to the way you interact with the world. This phenomenon, known as cortical remapping, means that your brain's physical structure is constantly being sculpted by your habits and skills. When a person spends years practicing the piano, the regions of the somatosensory cortex dedicated to their fingers actually expand, recruiting neighboring neurons to enhance their dexterity and tactile sensitivity. This adaptability is the secret behind the human ability to master complex tools until they feel like natural extensions of the body. Have you ever noticed how, after using a specific tool for a long time, you can almost feel the texture of what the tool is touching as if the sensation were moving through your own skin? This happens because the brain's map is flexible enough to incorporate external objects into its sensory schema. However, this remarkable plasticity can sometimes result in a strange biological echo known as phantom limb syndrome. When a person loses a limb, the corresponding section of the brain's map doesn't simply go dark, it remains hungry for input. In the absence of signals from the missing hand or leg, the surrounding neural territories, such as those representing the face, may begin to migrate into the vacant space. Because these regions are now overlapping, a simple touch on the cheek can trigger a vivid, ghostly sensation in a hand that is no longer there. It is a haunting reminder that our sense of physical presence is a sophisticated simulation, a masterpiece of biological mapping that continues to tell the story of the body even when the body itself has changed. This persistent map ensures that your internal sense of self remains coherent and whole, acting as the silent architect of your physical reality. Imagine a world where the flavor of a peach is not just a sweetness on the tongue, but the distinct shape of a cold, smooth sphere, or where the sound of a violin produces ripples of deep burgundy across your field of vision. This sensory overlap is known as synesthesia, a condition where the stimulation of one cognitive pathway leads to involuntary, automatic experiences in a second, unrelated pathway. For most of us, the brain keeps the senses in separate, neatly organized compartments, but for a synesthete, those boundaries are beautifully porous. This isn't a product of an overactive imagination. Rather, it is a fundamental difference in the architecture of the mind. Current theories in developmental neuroscience, such as those discussed in the journal Nature Reviews Neuroscience, suggest that we might all be born with these interconnected pathways. In infancy, the human brain is a dense thicket of neural connections, many of which are eventually pruned away as we grow and our brains become more specialized. In the minds of those who experience blended senses, it appears that some of these original bridges remain intact, allowing different regions to communicate in ways that are typically restricted. This crosstalk means that the visual cortex might activate not just in response to light, but in response to sound or even abstract concepts like time and mathematics. One of the most frequently documented forms is grapheme color synesthesia, where letters or numbers are inherently tied to specific hues. To someone with this trait, the letter A might always be a brilliant crimson, while the number 7 feels like a cool forest green. Have you ever noticed yourself associating certain days of the week with a particular color or a specific personality? Other individuals experience lexical gustatory synesthesia, where spoken words trigger specific tastes. A name might taste like tart apples, or a common noun might evoke the sensation of warm buttery toast. These associations are remarkably consistent over many decades, proving they are hardwired into the individual's neural map. This phenomenon offers a profound window into the nature of human consciousness, suggesting that our perception of reality is far more subjective than we often realize. If two people can look at the same black ink on a page and one perceives a vibrant rainbow while the other sees only monochromatic lines, it reveals that the brain is not a passive recorder of the world. Instead, it is an active creator of a private simulation. The hidden power of the brain lies in this ability to synthesize data into a coherent, albeit unique, internal theater. It reminds us that what we perceive as the solid, objective world is actually a masterful, internal construction, tailored by the specific wiring of our own neural pathways. This intricate lattice of connections isn't a static map established at birth and locked away in a vault. Instead, the human brain possesses a remarkable capacity for physical transformation known as neuroplasticity, which allows it to reorganize its very architecture in response to every thought, emotion, and environmental shift you experience. This ability means that your brain is more like a dynamic, living sculpture than a rigid computer hard drive. Within this internal landscape, a fundamental principle governs growth. Neurons that fire together, wire together. This concept, formalized by neuropsychologist Donald Hebb in 1949, describes how repeated signaling between specific cells strengthens the chemical bond at the synapse. When you learn a new skill or memorize a melody, your brain is actually reinforcing these pathways, making future communication more efficient. This specific type of change is called synaptic plasticity, focusing on the strength and volume of the signals sent between individual neurons. It involves the modulation of neurotransmitter release and the sensitivity of the receiving receptors to ensure the message is received clearly. On a larger scale, we observe structural plasticity, where the brain physically alters its shape by growing new dendrites or pruning away unused connections. This isn't just a metaphor. Studies using high-resolution imaging have shown that the physical volume of specific brain regions can increase through dedicated practice. For instance, researchers at University College London found that the posterior hippocampus, the area responsible for spatial navigation, actually grew larger in taxi drivers as they memorized the complex knowledge of the city's streets. Can you remember a time when a difficult task suddenly became second nature, almost as if your hands knew what to do before your mind caught up? This sensation is the physical manifestation of your motor cortex remapping itself to accommodate a new habit. This superpower becomes even more evident when we look at the brain's resilience following a traumatic injury or a stroke. When a specific region is damaged and can no longer perform its function, the brain often initiates a process called functional reorganization. Healthy, adjacent areas of the cortex can eventually take over the tasks once managed by the injured tissue, effectively rerouting the neural traffic through new territory. This recovery highlights the brain's inherent drive to maintain its operational integrity even when faced with significant physical setbacks. A protein called brain-derived neurotrophic factor, or BDNF, acts like a fertilizer in this process, supporting the survival of existing neurons and encouraging the growth and differentiation of new ones. While scientists once believed that the adult brain was fixed or hardwired, we now know that neurogenesis, the birth of new neurons, continues throughout life, particularly in the dentate gyrus of the hippocampus. This region is vital for forming new memories and regulating emotions. The brain also employs a process called synaptic pruning, which is especially active during childhood and adolescence, where it systematically eliminates weaker connections to make the overall network more streamlined and energy efficient. It is a use it or lose it system that ensures the most important pathways remain strong and responsive. Myelination also plays a key role here, as the brain wraps frequently used axons in a fatty insulation to speed up electrical impulses. This coating can increase the speed of information travel by up to 100 times compared to unmyelinated fibers. Every time you engage in a deep, focused activity, you are essentially signaling to your brain that these specific pathways are worth the investment of more myelin. This continuous refinement ensures that your internal world remains perfectly tuned to the demands of your external life. As we navigate the world, the brain operates much like a sculptor working with living, adaptive clay. When you first attempt a new task, perhaps learning to play a stringed instrument or mastering a complex software, your prefrontal cortex is under immense metabolic strain. This initial stage, often called conscious competence, requires your full attention because the neural pathways involved are still fresh and unrefined. You feel every mistake and every hesitation because the electrical signals are traveling along relatively slow, uninsulated circuits. However, as you repeat these actions, a physical transformation begins within the white matter. The brain starts to deposit more layers of myelin around the active axons, a process that functions like upgrading a dial-up connection to high-speed fiber optics. This thickening of the myelin sheath allows the electrical pulses to travel with incredible velocity and precision, which is the biological reality behind what we often call muscle memory. Interestingly, your muscles do not actually possess memory. Instead, the neural signals controlling them have simply become so efficient that they no longer require your conscious mind to intervene. Science further categorizes these achievements into two distinct systems: declarative and procedural memory. Declarative memory is our storage for facts, names, and events, the kind of information we can declare or speak out loud. This relies heavily on the hippocampus, which acts as a sort of staging ground for new data. In contrast, procedural memory is the how-to of our lives, stored deep within the basal ganglia and the cerebellum. These two systems are so independent that they can function even when the other is damaged. Think of a professional golfer who can still swing a club with perfect form even if they cannot remember the name of the course they are playing on. Can you remember a time when a skill you practiced for hours suddenly felt like it was happening on its own? This shift marks the transition into subconscious mastery, where the brain moves the workload from the energy-intensive outer layers to the more efficient, automated deeper structures. While we often think of learning as adding new connections, the brain's true efficiency comes from a process known as synaptic pruning. During our younger years, our brains are a dense, tangled forest of synaptic connections, many of which are redundant or inefficient. As we specialize in certain skills or environments, the brain identifies the pathways that are rarely used and systematically dismantles them. This biological streamlining is not a loss. It is a sophisticated optimization. By removing the noise of unused connections, the brain ensures that the signal of our most important skills travels without interference. This is why an expert's brain actually shows less activity on a scan than a beginner's during the same task. The expert's brain has been pruned and insulated into a high-performance machine that achieves more while consuming less energy. This constant refinement means that your brain is a living record of your persistence, physically reshaped by the specific ways you choose to engage with the world around you. Every hour of focused practice is a signal to this internal architect to continue the work of building a more efficient version of yourself, ensuring that your cognitive resources are never wasted on pathways that no longer serve your goals. This biological construction begins with an explosive period of growth that is unmatched at any other stage of life. In the first few years after birth, the brain forms upwards of 1 million new neural connections every single second. While a newborn enters the world with almost all the neurons they will ever possess, the intricate web of synapses, the points of communication between these cells, is only just beginning to weave itself together. By the age of 3, a child's brain has roughly 1 quadrillion synaptic connections, which is nearly double the amount found in a typical adult. This period of exuberant growth creates a brain that is hypersensitive to its surroundings, allowing a child to absorb the complexities of their culture and environment with startling efficiency. Nature manages this surplus of connectivity through a ruthless but necessary process known as synaptic pruning. Think of it as a sculptor removing excess stone to reveal the statue within. The brain identifies which pathways are used most frequently and reinforces them, while those that remain dormant are systematically dismantled to conserve energy and improve processing speed. This streamlining is particularly evident during critical periods, which are specific developmental windows where the brain is uniquely primed to acquire certain skills. Vision, for example, requires specific environmental input during the first few months of life to calibrate the neural circuits responsible for depth perception and color recognition. If these inputs are missing, the brain may permanently reallocate that neural real estate to other senses. Do you have a vivid memory of a specific melody or a storybook from when you were very small? Language acquisition operates on a similar biological schedule, with the brain's plasticity for phonetic sounds peaking before the age of 7. During this window, a child can distinguish between the sounds of every language on Earth, a capability that gradually diminishes as the brain specializes in the specific dialect it hears most often. As the child transitions into adolescence, the focus of development shifts toward the reorganization of the frontal lobes. The teenage brain is often characterized by a specific developmental lag that influences behavior in profound ways. While the limbic system, the region responsible for emotions and the pursuit of rewards, becomes highly active during puberty, the prefrontal cortex remains under construction. This part of the brain acts as the executive control center, managing impulses, assessing risks, and planning for the future. Because the emotional engine matures faster than the cognitive brakes, adolescents often experience the world with heightened intensity and a biological predisposition toward novelty seeking. This is not a flaw in design, but rather a vital stage that encourages independence and exploration. Current neuroscientific research, including longitudinal studies from the National Institutes of Health, confirms that this maturation process is much slower than previously thought. The human brain does not reach its full structural and functional maturity until the mid-20s, as the final layers of myelin are wrapped around the long-range fibers that connect our emotional centers to our rational mind. Once this peak of development is reached, the narrative of the brain often shifts toward a story of inevitable decline, yet the biological reality is far more resilient. Our neural architecture possesses a remarkable ability to reorganize itself through a phenomenon known as cognitive reserve. This concept suggests that the brain is not a rigid piece of hardware, but a dynamic system capable of building backup pathways. When certain neurons or connections begin to falter with age, the brain draws upon these secondary networks to maintain its function. Have you ever noticed how your ability to recognize complex social patterns or solve nuanced problems seems to have deepened over the decades? This is because while raw processing speed, the sheer velocity at which we can react to a simple stimulus, tends to peak in our early 20s, other forms of intelligence continue to climb. The aging brain often adopts a strategy called bilateral activation, where it recruits regions from both the left and right hemispheres to complete a task that a younger brain would handle with only one side. This cooperative effort allows the mind to stay sharp even as individual cells are lost. Even more surprising is the discovery of adult neurogenesis, particularly within the hippocampus, the region dedicated to memory and spatial navigation. For a long time, science held the consensus that we were born with all the neurons we would ever have, but we now know that the brain continues to birth new cells well into our 80s and 90s. These newborn neurons integrate into existing circuits, helping to sharpen our ability to distinguish between similar memories. The brain also relies on its vast library of stored experiences, moving away from the frantic energy of youth toward a more efficient use of pattern recognition. This is often described as the transition from fluid intelligence to crystallized intelligence. While the young brain is fast, the mature brain is wise, utilizing a lifetime of accumulated data to reach conclusions through intuition and synthesis that a younger mind might miss. This structural adaptability, often referred to as the scaffolding theory of aging and cognition, ensures that the brain remains a work in progress, constantly refining its methods to meet the challenges of time. Research into neuroplasticity shows that engaging in cognitively demanding tasks, such as learning a new language or mastering a complex craft, physically strengthens the white matter tracts that facilitate communication between distant brain regions. This ongoing maintenance turns the brain into a living archive, one that prioritizes the depth of understanding over the speed of execution. Beyond the neurons themselves, support cells known as microglia and astrocytes play a critical role in this late-stage optimization, clearing out metabolic waste and repairing minor damage to the myelin sheaths. The brain's ability to work around physical changes is so profound that many individuals exhibit the physical hallmarks of aging under a microscope while maintaining perfectly clear and vibrant mental faculties in their daily lives. By focusing on the strength of the whole network rather than the health of individual cells, the human brain reveals its most enduring superpower, the ability to endure, adapt, and even thrive as the years pass. This graceful transition into the later stages of life is less about loss and more about a sophisticated refinement of how we perceive the world. The transformation of a fleeting moment into a permanent part of our identity begins with the intricate machinery of memory, starting with the delicate act of encoding. This is the biological gateway where the external world is translated into an internal reality. Before any piece of information can be tucked away for a lifetime, it must navigate three distinct phases: encoding, storage, and finally, retrieval. Think of encoding as the brain's diligent scribe, taking the raw data of a scent, a sound, or a conversation and converting it into a chemical and electrical language the neurons can understand. Without this initial translation, our experiences would wash over us like water over stone, leaving no trace behind. This process ensures that the brain does not just react to the world, but actually learns from it. The primary catalyst for this entire system is the power of attention. If the human brain is a vast, quiet library, attention is the spotlight that determines which book the scribe actually reads. Every second, our senses are bombarded with millions of bits of data, yet we only encode a tiny fraction of what we encounter. This selective filtering is a biological necessity. If we encoded every stray noise or flicker of light, our neural networks would quickly become overwhelmed by noise. Do you ever find yourself reaching the bottom of a page only to realize your mind was elsewhere, and you have no memory of the words you just saw? This occurs because the physical pathways required for encoding were never fully activated, leaving the information to evaporate before it could be processed. Once information passes through the filter of attention, it enters the brief, shimmering realm of short-term memory. In the world of neuroscience, this is frequently called working memory, a term that better describes its role as a mental workspace where we hold and manipulate information. This is where the brain performs its most immediate tasks, such as holding a temporary address in mind while navigating a car. A famous study by cognitive psychologist George Miller in the 1950s identified what he called the magic number 7, suggesting that the average human can hold roughly 7 items, plus or minus 2, in their working memory at any given time. While more recent research suggests the limit might be slightly lower for complex data, this fundamental constraint shapes how we interact with the world. To overcome these biological limits, the brain utilizes a sophisticated technique known as chunking. By grouping individual pieces of data into larger, more meaningful units, we can effectively expand the capacity of our working memory without changing its physical structure. A random string of 12 letters is nearly impossible for most people to remember instantly, but if those letters form 3 familiar words, the brain treats those words as 3 single chunks, making the information easy to retain. These relaxing facts about our cognitive shortcuts illustrate how the brain maximizes efficiency, allowing us to process complex ideas during bedtime listening without overtaxing our mental resources. The transition from these temporary states to a permanent record is managed by the hippocampus. Tucked deep within the temporal lobe and shaped somewhat like a seahorse, the hippocampus serves as the save button of the brain. It does not store the memories itself, rather, it acts as a conductor, directing the information to various parts of the cerebral cortex for long-term storage. This transition is a process called consolidation, where the brain strengthens the synaptic connections between neurons. If the hippocampus is damaged, a person may retain all their old memories but lose the ability to form a single new one, living in a permanent, unchanging present. During consolidation, the brain employs a mechanism known as long-term potentiation, which is the persistent strengthening of synapses based on recent patterns of activity. This is the physical realization of the scientific law that neurons which fire together wire together. As the body rests, the hippocampus is busy sorting through the day's encoding, deciding which signals are worth the energy of a permanent structural change and which can be discarded. This is why science for sleep is such a compelling field. It reveals that even in our quietest moments, the brain is actively sculpting its own physical architecture to preserve the essence of our experiences. Every new piece of knowledge subtly alters the brain, ensuring that our internal landscape is constantly expanding and refining itself. The sheer volume of data the human mind can hold is staggering, often estimated by neuroscientists to be in the range of several petabytes. To put that into perspective, if your brain functioned like a digital video recorder, it could store roughly 3 million hours of high-definition footage, enough to play continuously for over 300 years. This incredible density comes from the way neurons branch and connect, with a single cubic millimeter of cerebral cortex housing nearly 1 billion synapses. Researchers at the Salk Institute discovered that these synapses are far more sophisticated than simple binary switches, as they can adjust their size and strength across 26 different levels. This variability essentially increases the storage capacity of each individual connection, allowing the brain to pack an unimaginable amount of information into its 3-pound structure. Yet, despite this massive potential, our memories do not function like archival film reels or static digital files waiting to be accessed. Every time we reach back into our past, the brain engages in a process of active reconstruction rather than simple playback. A memory is less like a photograph and more like a theatrical performance, where the brain gathers various threads of information and weaves them together anew each time they are summoned. This fluid nature of recall explains why our personal histories can shift and evolve as we age, reflecting our current understanding of the world. Perhaps even more fascinating is the way these memories are physically distributed throughout the mind, a concept often explored through the lens of holographic theory. Early neurological experiments by Carl Lashley revealed that removing specific parts of a brain did not necessarily erase a specific learned behavior, suggesting that knowledge is not stored in one isolated cabinet. It appears that the essence of an experience is smeared across vast networks of the cortex, meaning that even if one area is damaged, the memory can often still be retrieved from the remaining fragments. This distributed architecture mirrors a hologram, where every small piece of the film contains the data for the entire image, providing a natural safeguard against the loss of vital information. Over time, these distributed patterns are reinforced through the synthesis of specific proteins, making long-term memories remarkably durable and resistant to decay. This biochemical hardening ensures that the faces of loved ones and the fundamental lessons of childhood can persist for many decades, anchored deeply within the physical fabric of the mind. Have you ever noticed how a specific scent can instantly transport you back to a moment from your early childhood? This enduring stability allows us to maintain a consistent sense of self throughout the changing chapters of our lives, as the brain meticulously preserves the core pillars of our identity while constantly updating the finer details of our daily experiences. Because these networks are so deeply integrated, the brain can prioritize the most relevant information, ensuring that while the storage is vast, the retrieval remains incredibly efficient. We often view the loss of a memory as a failure of our biological hardware, yet forgetting is actually an active, sophisticated metabolic process essential for mental clarity. Rather than a passive decay, the brain utilizes specific proteins and signaling pathways to intentionally erase information that no longer serves a direct purpose. This prevents a state of synaptic saturation, where the sheer volume of neural connections would create too much background noise for the system to function effectively. If every mundane detail of your life remained permanently accessible, your ability to make decisions or identify patterns would be paralyzed by the clutter of irrelevant data. Biological research suggests that the protein RAC1 helps manage this process by mediating the restructuring of the cytoskeleton within synapses, effectively wiping the connections that haven't been reinforced. Scientists refer to this pruning as a way of maintaining the brain's plasticity, ensuring that new, more vital information always has a place to land. Have you ever noticed how some memories from childhood remain vivid while what you ate for lunch 3 days ago has completely vanished? This selective erasure is beautifully illustrated by the forgetting curve, a concept pioneered by psychologist Hermann Ebbinghaus, which shows how information leaves our consciousness at an exponential rate unless it is actively reinforced. Spaced repetition acts as the natural counterforce to this decline, signaling to the hippocampus that a particular piece of data is worth the metabolic cost of long-term storage. When we encounter information at increasing intervals, the brain strengthens those specific synaptic pathways, effectively shielding them from the daily cleanup crew. However, the brain does not treat all data equally, as it possesses a built-in hierarchy for what stays and what goes. Information tied to strong emotional states or survival needs is fast-tracked for preservation, often bypassing much of the standard filtering process. The amygdala works in tandem with the memory centers to tag these experiences with high priority, ensuring that a dangerous encounter or a significant life milestone is etched deeply into our neural architecture. This creates a functional trade-off where the brain sacrifices the mundane to safeguard the meaningful. By clearing out the background noise of daily existence, the mind creates a streamlined environment where high-value information can be retrieved with lightning speed. This biological housekeeping is what allows us to adapt to a changing world, rather than being stuck in a static loop of every sensation we have ever experienced. In this way, the superpower of memory is actually dependent on the equally powerful, silent work of forgetting, which keeps the cognitive machine lean and focused on the present moment. While we are conscious, the brain is a furnace of activity, consuming roughly 20% of the body's total oxygen and glucose despite making up only 2% of its weight. This intense metabolic fire produces a significant amount of molecular soot that must be dealt with to prevent cognitive smog. For over a century, the mechanism for this disposal remained one of biology's greatest mysteries because the brain lacks the lymphatic vessels found elsewhere in the body. In 2012, researchers at the University of Rochester discovered that the brain compensates for this by turning its entire structure into a temporary filtration plant. The glymphatic system acts as a high-pressure rinse, utilizing specialized channels formed by astrocytes to guide cerebrospinal fluid through the deep tissues. This plumbing network remains mostly silent during the day, waiting for the shift in consciousness that signals it is safe to begin the cleaning process. During our waking hours, the brain is too busy processing the world to focus on sanitation, keeping its cells plump and pressed tightly together. Once sleep takes hold, a remarkable transformation occurs as the space between these cells expands by up to 60%, creating wide corridors for the cleaning fluid to flood the organ. This surge of fluid doesn't just flow passively, it is propelled by the rhythmic pulsing of the brain's arteries, which act like mechanical pumps to drive the fluid forward. Do you prefer a cool room or a warm one when you are settling in for the night? Temperature regulation is closely tied to this process, as the brain must reach a specific thermal state to facilitate the most efficient waste clearance. When the system is firing on all cylinders, it effectively strips away the chemical byproducts of a long day of thinking. This nightly wash targets specific proteins like beta amyloid, which are known to clump together and form plaques if left undisturbed. By flushing these toxins into the circulatory system for eventual disposal by the liver, the glymphatic system acts as a primary defense against cognitive decline and neurological disease. When we choose to stay awake, we are essentially refusing to let the janitorial crew enter the building. The resulting brain fog is the physical manifestation of metabolic trash clogging the synapses, slowing the speed of electrical signals and dulling the sharpness of our perceptions. This explains why the clarity of a well-rested mind feels like a superpower compared to the sluggishness of exhaustion. Beyond mere cleaning, this system also helps distribute essential nutrients like glucose, lipids, and amino acids to the far reaches of the cortex. It is a dual-purpose logistics network that ensures the brain is both nourished and sanitized before the sun rises. Energy conservation theories suggest that the brain simply cannot afford to run its high-level cognitive functions and its deep cleaning protocols at the same time. The metabolic cost of moving fluid through the dense tissue of the brain is so high that it requires a total shutdown of external awareness. By viewing sleep through this lens, we see it not as a luxury or a period of lost time, but as a vital biological necessity for maintaining the integrity of the mind's physical structure. This hidden internal maintenance is what allows the brain to wake up each morning with its vast potential fully restored. Each night of deep, undisturbed rest is a gift to your future self, ensuring that the delicate architecture of your memories and personality remains clear of the debris that naturally accumulates from the simple act of being alive. As the night deepens, the brain shifts its focus from structural maintenance to the complex management of information and identity. Within the theater of the mind, rapid eye movement sleep emerges as a sophisticated laboratory for emotional processing. During REM, the brain is nearly as active as it is when we are wide awake, yet it is neurochemically unique. The brain's stress-related chemical, noradrenaline, is completely shut off during this stage, allowing us to process traumatic or challenging experiences in a chemically calm environment. This mechanism functions as a form of overnight therapy, stripping away the raw, painful edge from memories so they can be stored as facts rather than recurring emotional triggers. While you rest, your amygdala, the center for processing emotions, is highly active, working in tandem with the hippocampus to decide which feelings are worth keeping and which can be safely diffused. While the emotional landscape is being leveled, a rigorous filing system is simultaneously at work. This process, known as memory consolidation, involves a high-speed dialogue between the hippocampus and the neocortex. Throughout the day, the hippocampus acts as a temporary storage site for new experiences, but its capacity is limited. During sleep, it replays these experiences, firing the same neural sequences that were active during the initial event. This replay occurs at a significantly accelerated pace, effectively teaching the neocortex what to remember. By repeating these patterns thousands of times in a single night, the brain carves deep, permanent channels into our long-term memory. It is a meticulous archival process that ensures the skills you practiced or the information you studied becomes a permanent part of your cognitive hardware. To prevent the body from physically reacting to these vivid mental rehearsals, a remarkable safety mechanism takes hold in the brainstem. The pons and the medulla send powerful inhibitory signals down the spinal cord, utilizing neurotransmitters like GABA and glycine to shut down motor neurons. This induces a temporary state of paralysis known as REM atonia. It is a protective superpower that ensures while you might be sprinting through a dream landscape or navigating a complex scenario, your physical limbs remain safely stationary in bed. Have you ever experienced that strange, heavy sensation of trying to run in a dream but feeling like your legs are moving through honey? This is often the conscious mind sensing the natural atonia that keeps the body still, a bridge between the vividness of the dream world and the safety of physical immobility. Dreams themselves serve as a bridge between the known and the unknown, allowing the brain to engage in a process called pattern matching. During the day, the prefrontal cortex keeps our thoughts linear and logical, but during REM, this area is less active, allowing for more divergent and creative associations. The brain uses these hours to connect new information with existing knowledge in ways that waking logic might never allow. This is why we often wake up with the solution to a complex problem or a sudden ah moment. The brain has been cross-referencing disparate data points all night. This hidden superpower of synthesis is what allows humans to find meaning and innovation in a chaotic world. Sometimes, the boundary between the conscious and the subconscious blurs in a phenomenon called lucid dreaming. This occurs when the dorsolateral prefrontal cortex, the seat of self-awareness and executive function, reactivates while the rest of the brain remains deep in a REM cycle. Suddenly, the dreamer becomes aware that the reality surrounding them is a mental construct. In this state, the brain exhibits a unique hybrid of waking and sleeping brainwave patterns, allowing the individual to consciously observe or even influence the dream's narrative. It is one of the few times we can witness the brain's creative machinery in real time, highlighting the incredible flexibility of human consciousness. Every minute spent in these cycles contributes to your cognitive resilience, ensuring that when you open your eyes, your mind is not just rested, but thoroughly updated and refined. While you navigate your daily life believing you are the primary architect of every choice, your biological hardware is often several steps ahead of your conscious awareness. Neuroscientific research, pioneered by Benjamin Libet in the 1980s and refined by modern fMRI studies at the Max Planck Institute, reveals that the motor cortex begins preparing for a movement several hundred milliseconds before you feel the urge to act. In some complex decision-making scenarios, patterns of neural activity can predict a choice up to 10 full seconds before it enters your stream of consciousness. This suggests that the feeling of deciding is not the beginning of the process, but rather a late-stage notification delivered to the conscious mind. The sheer scale of data managed beneath the surface is staggering when compared to the narrow bandwidth of our focused attention. To quantify this, scientists estimate the conscious mind processes roughly 40 to 50 bits of information per second, a capacity roughly equivalent to reading a single short sentence. Meanwhile, the subconscious systems are handling an estimated 11 million bits per second, managing everything from internal homeostatic regulation to the silent interpretation of peripheral visual cues. This massive discrepancy exists because the brain is an organ optimized for survival rather than constant self-reflection. If you had to consciously calculate the trajectory of every moving object or the syntax of every spoken word, your cognitive resources would vanish instantly under the weight of the data. To maintain this efficiency, the brain employs cognitive shortcuts known as heuristics, which function as mental rules of thumb to bypass exhaustive analysis. These shortcuts allow for rapid-fire judgments that preserve the body's precious glucose and oxygen supplies, as the brain consumes 20% of your total energy despite making up only 2% of your mass. One common mechanism is the availability heuristic, where the mind prioritizes information that is most easily recalled, often mistaking ease of memory for frequency of occurrence. Another is the anchoring effect, where your initial exposure to a specific number or value subtly biases every subsequent judgment you make, regardless of its relevance. These patterns are not flaws, but rather highly evolved strategies designed to produce good enough results in environments where speed is often more valuable than perfect accuracy. This leads to the realization that much of what we perceive as deliberate agency is actually a form of retrospective storytelling. When the subconscious mind makes a snap judgment, the conscious mind often creates a rationalization after the fact to maintain the illusion of total control, a phenomenon known in psychology as confabulation. We observe this in patients with split brain conditions who, when their right hemisphere is given an instruction, will invent a logical reason for the action using their left hemisphere, unaware of the true trigger. Have you ever noticed yourself reacting to a sudden sound or movement before you even realized what it was? It creates a seamless narrative of the self, allowing you to move through the world with a sense of unified purpose even as your underlying neural circuits operate in a decentralized, parallel fashion. This protective delay is a testament to the silent, vigilant guardian that resides within your cranium, constantly evaluating threats and opportunities long before you have the chance to think about them. The conversation between your mind and your body extends far beyond the borders of the skull, reaching down into the core of your torso through a complex biological highway. This communication channel is anchored by the vagus nerve, a wandering bundle of fibers that serves as the primary conduit for the gut-brain axis. While we often think of the brain as the sole commander of the body, the relationship between the cranium and the digestive tract is surprisingly democratic. Approximately 80% of the signals traveling through the vagus nerve are actually ascending, moving from the gut upward to the brain, rather than the other way around. This constant stream of data provides the brain with a real-time report on the internal state of the body, influencing your subconscious sense of well-being. Nestled within the walls of your digestive system is a vast network of neurons known as the enteric nervous system, which is so extensive that scientists frequently refer to it as the second brain. It operates with a startling level of independence, managing the complexities of digestion and nutrient absorption without needing direct instructions from the head. This second brain contains more than 100 million neurons, which is a greater number than those found in either the spinal cord or the peripheral nervous system. It is here that some of the most influential chemical messengers in the human body are manufactured in surprising quantities. You might be surprised to learn that about 95% of the body's serotonin, a neurotransmitter essential for regulating mood and sleep, is produced in the gut rather than the brain. These chemical signals are generated by specialized cells in the intestinal lining and are influenced heavily by the trillions of microbes living within you. This community of bacteria, known as the microbiome, functions almost like a hidden organ with its own metabolic and signaling capabilities. Specific strains of bacteria are capable of producing other critical neuroactive substances, including gamma-aminobutyric acid, or GABA, which helps the brain manage anxiety and maintain a state of calm. When you experience a sudden gut feeling or a sense of butterflies in your stomach during a moment of nervousness, you are witnessing a literal biological event where the brain and the enteric nervous system are in rapid dialogue. This is not merely a poetic metaphor. It is the physical manifestation of the gut-brain axis responding to stress or anticipation. Research published in journals like Nature and Cell suggests that the health of this microbial ecosystem can directly alter the structural integrity of the brain's emotional centers, such as the amygdala and the hippocampus. The composition of your gut flora can even dictate how you respond to social stress. Do you remember a time when a simple meal seemed to completely shift your outlook on a difficult day? This phenomenon occurs because the microbiome communicates with the brain via the immune system, the endocrine system, and the vagus nerve simultaneously. When the gut is in a state of dysbiosis or imbalance, it can send signals of distress that the brain interprets as anxiety or low mood. Conversely, a diverse and healthy microbiome produces short-chain fatty acids that protect the blood-brain barrier and support the growth of new neurons. This silent partnership ensures that your mental clarity and emotional resilience are deeply rooted in the biological health of your digestive system, proving that the mind is a whole-body experience. Deep within the primitive architecture of the skull lies the brainstem, a tireless sentinel that functions as the body's master autopilot. While your conscious thoughts might be occupied with complex problems or drifting into the quietude of sleep, this ancient structure never wavers in its management of the vital signs that keep you alive. It regulates the steady beat of your heart and the rhythmic expansion of your lungs with such precision that you rarely need to give them a second thought. This silent governance is handled by the autonomic nervous system, a sophisticated internal network divided into two distinct branches that exist in a state of perpetual, dynamic balance. The sympathetic nervous system functions much like an accelerator, preparing the body for immediate action by dilating the pupils and surging blood toward the large muscle groups. This fight-or-flight mechanism was once our primary defense against environmental threats, and even today, it remains a marvel of rapid biological mobilization. In direct opposition stands the parasympathetic nervous system, the body's natural braking system, often referred to as the rest and digest mode. This branch is responsible for the quiet work of cellular repair, lowering the heart rate and facilitating the complex chemical processes of digestion and immune function. The harmony between these two systems determines your overall resilience to the world around you. However, modern life often keeps the sympathetic branch in a state of overactivation, leading to a phenomenon where stress effectively hijacks the prefrontal cortex. This part of the brain, usually reserved for logical decision-making and executive function, becomes less active as the survival-oriented amygdala takes the lead. When this happens, your ability to think creatively or remain objective is physically suppressed by your own neurochemistry. Have you ever felt your mind go blank during a moment of high pressure, only to have the answers return once you've finally calmed down? This is the result of the brain shifting its metabolic resources to the areas it deems most critical for survival at that moment. Despite the seemingly involuntary nature of these processes, we are not entirely at the mercy of our biology. Through the practice of biofeedback, researchers have shown that humans can learn to exert conscious influence over their autonomic functions. By observing real-time data of their own heart rate or brain waves, individuals can develop the skill to lower their blood pressure or alter their internal temperature through mental focus. This reveals that the bridge between the conscious mind and the autonomic system is much wider than we once suspected, offering a profound level of agency over our physiological state. By understanding the pathways of the vagus nerve, which acts as the primary conduit for parasympathetic signals, we can essentially hack our own nervous system to restore internal calm. This internal regulation extends into one of the most sophisticated functions of the human mind, the construction and modulation of pain. It is a common misconception that pain is a simple signal traveling from a wound directly to the consciousness, like electricity through a wire. In reality, pain is an elaborate output created by the brain, a calculated opinion on the safety of the body rather than a direct measurement of tissue damage. The physiological process begins with nociception, where specialized nerve endings detect mechanical, thermal, or chemical threats. These sensors send raw data toward the spinal cord, but this data is not yet pain. It is merely information. Whether that information becomes a sensation of suffering depends entirely on the brain's interpretation of the current environment. This filtering process is best explained by the gate control theory of pain, which suggests that the spinal cord contains a neurological gate that either blocks pain signals or allows them to continue to the brain. When you instinctively rub a stubbed toe, you are utilizing this mechanism. The tactile pressure from your hand sends fast-moving sensory signals that effectively crowd out the slower pain messages, closing the gate and dampening the intensity of the discomfort. Do you remember a time when you discovered a bruise or a scratch on your arm but had no memory of how it happened? This occurs because the brain can selectively mute incoming nociceptive signals when it is occupied with more pressing matters. In survival mode or high-stakes environments, the brain prioritizes action over awareness of injury. An athlete may finish a game on a broken limb, or a soldier might ignore a wound until safety is reached, because the brain's descending inhibitory pathways flood the system with endogenous opioids like endorphins and enkephalins. These are the brain's own internal pharmacy, capable of producing effects as potent as morphine to ensure the body remains functional during a crisis. The complexity of this superpower is revealed in the distinction between the sensory discriminative aspect of pain, where it is and what it feels like, and the effective motivational component, which is how much the pain bothers us. These two experiences are processed in different regions of the brain. While the somatosensory cortex identifies the location of a pinch, the anterior cingulate cortex and the insula determine the emotional weight of that pinch. Because these systems are distinct, the brain can actually decouple the sensation from the suffering. This is why context matters so much. A burn from a hot stove feels agonizing, yet the same level of heat in a therapeutic sauna might feel relaxing. The brain evaluates the threat level and adjusts the volume of the signal accordingly. However, this system can sometimes become oversensitized, leading to chronic pain states where the brain continues to produce a pain response long after the physical tissue has healed. In these instances, the nervous system has essentially learned to be in pain, demonstrating the incredible, if sometimes counterproductive, plasticity of our neural circuits. By understanding that the brain is the ultimate architect of our sensory reality, we begin to see how psychological states, expectations, and even the focused direction of our attention can fundamentally rewrite our physical experience of the world. This capacity to modulate the most intense of human sensations proves that we are not merely passive recipients of biological signals, but active participants in the creation of our own perceived existence. This internal pharmacy operates through the complex mechanics of expectation, where the simple belief in a remedy triggers the brain to synthesize its own potent medicinal compounds. When you anticipate relief from pain, your prefrontal cortex signals the periaqueductal gray area of the midbrain to release endogenous opioids. These are naturally occurring chemicals, like endorphins and enkephalins, that possess a molecular structure strikingly similar to morphine. They flood your system, physically docking with opioid receptors in the spinal cord to inhibit the upward flow of pain signals before they ever reach conscious awareness. This process is not a psychological trick or a lapse in judgment, but a measurable biochemical event where the brain preemptively alters the body's internal chemistry to match a predicted outcome. Brain imaging studies using PET scans have demonstrated that patients experiencing the placebo effect show increased dopamine activity in the striatum, a region associated with reward and motivation. This suggests that the mere promise of recovery acts as a biological catalyst, prompting the brain to invest its own chemical resources into the healing process. Have you ever noticed how the mere act of sitting in a doctor's waiting room can sometimes make your symptoms feel less intense? This phenomenon extends beyond pain management into the regulation of the immune system and motor functions. In individuals with Parkinson's disease, for example, the expectation of a treatment can stimulate the release of dopamine in the brain, temporarily improving motor control through purely internal mechanisms. However, this neurological power is a double-edged sword, manifesting in the darker twin of healing known as the nocebo effect. If the placebo effect is the biology of hope, the nocebo effect is the chemistry of dread. When a person expects a specific side effect or fears a certain substance, the brain can generate genuine physical symptoms in response to that negative anticipation. This is driven largely by the release of cholecystokinin, a peptide that facilitates pain transmission and heightens anxiety. The nocebo response can cause blood pressure to spike, digestive systems to falter, and skin rashes to appear, all fueled by the brain's interpretation of a perceived threat. It reveals a profound vulnerability in our biology. The brain does not always distinguish between a physical toxin and the mental certainty of being poisoned. This suggests that our physiological state is constantly being calibrated by a sophisticated predictive model that prioritizes our expectations over raw sensory data. The brain acts as a central governor, deciding which signals to amplify and which to suppress based on what it believes is about to happen. This creates a feedback loop where the mind's narrative dictates the body's reality, proving that the boundary between mental and physical is far more porous than we once believed. Every thought carries a chemical signature, and every expectation sets a biological stage, turning the brain into an active architect of our physical health and sensory experience. Within the architecture of the human brain, there exists a specialized class of cells that act as a bridge between the self and the other, dissolving the isolation of the individual mind. These mirror neurons represent a biological mechanism for connection, allowing one person to internally simulate the actions, intentions, and even the emotions of another. This internal mirroring extends far beyond simple motor tasks, reaching deep into the emotional core of our social interactions and creating a shared neural space. When we witness a friend stub their toe or a stranger display a look of pure joy, our own brains activate in patterns that nearly mirror their internal state. This phenomenon, known as neural resonance, relies heavily on mirror neurons located within the premotor cortex and the inferior parietal lobule. These cells do not merely process visual information, they translate the movements and expressions of others into our own internal language of sensation. It is as if the brain is constantly running a quiet, sophisticated simulation of the lives unfolding around it, turning observation into a form of lived experience. This capacity for simulation is what allows for the profound experience of empathy, where the line between your suffering and mine becomes biologically blurred. Researchers at the University of Parma first stumbled upon these neurons in macaque monkeys during the early 1990s, noticing that the same brain cells fired whether a monkey grasped a piece of food or simply watched a researcher do the same. In humans, this system has evolved into a complex network that supports social bonding and collective understanding through high-fidelity mimicry. This neural resonance explains why we might find ourselves flinching during a tense scene in a film or feeling a genuine lump in our throat when someone else cries. The anterior insula and the anterior cingulate cortex are particularly active during these moments of emotional mirroring, specifically when we observe others experiencing pain or disgust. The brain actually recruits the same sensory regions it would use if the physical trauma were happening to its own body. This shared activation forms the bedrock of effective empathy, ensuring that the social group remains sensitive to the needs and states of its individual members. One of the most common and puzzling manifestations of this system is the phenomenon of contagious yawning. While scientists have proposed various theories for why yawning spreads, including the regulation of brain temperature, the prevailing consensus points toward a deep-seated social function. Contagious yawning appears to be an involuntary sign of social rapport, showing that our brains are instinctively attuned to the physiological rhythms of those nearby. Studies using fMRI have shown that the posterior cingulate cortex and the precuneus activate during these moments, linking the yawn to the brain's social processing centers. Interestingly, this response is often absent in very young children or in individuals with conditions that affect social processing, suggesting it is a developmental milestone of a healthy, empathetic social brain. From an evolutionary perspective, this mirroring provides a massive survival advantage. It allowed our ancestors to predict the intentions of predators or rivals before a single move was made. By simulating the motor plans of others, the brain can anticipate what happens next, turning a social group into a single, cohesive unit capable of reacting with lightning speed. This shared neural space facilitates the transfer of skills through simple observation, bypassing the need for complex verbal instruction. Whether it is learning to use a tool or understanding the subtle cues of a tribal hierarchy, mirror neurons act as the silent teachers of the human experience. They turn the brain from an isolated processor into a resonant chamber tuned to the frequency of the human collective. This ability to feel what another feels is a hardwired biological reality that ensures our survival as a social species, making the social world a predictable and shared environment. Beneath the surface of every casual conversation lies a sophisticated biological network dedicated to the architecture of meaning. This specialized machinery centers largely within the left hemisphere of the brain, where two primary hubs orchestrate the symphony of human communication. Broca's area, tucked into the frontal lobe, serves as the primary engine for speech production, meticulously coordinating the muscle movements of the mouth and larynx. Just behind it, nestled in the temporal lobe, sits Wernicke's area, which acts as the brain's chief translator for incoming sound. While Broca's area builds the sentences, Wernicke's area decodes the patterns, ensuring that the vibrations hitting your eardrum transform into coherent concepts. These two regions are physically linked by a bundle of nerve fibers known as the arcuate fasciculus, a neural highway that allows for the near-instantaneous loop of listening and responding. The sheer speed of this system is staggering, as the brain typically begins formulating a response to a sentence before the speaker has even finished their thought. This predictive processing allows human dialogue to flow with a rhythm that feels effortless despite the astronomical number of calculations occurring behind the scenes. Translating a silent, abstract thought into a structured sentence requires the brain to navigate a complex web of syntax and morphology. Before a single word is spoken, the brain must select the correct lexical items from a mental dictionary containing tens of thousands of entries and then arrange them according to rigid grammatical rules. This process involves the basal ganglia, which helps sequence the timing of speech, ensuring that syllables emerge in the correct order. For those who speak more than one language, the brain employs an even more impressive superpower known as executive control to manage multiple linguistic systems simultaneously. Multilingual individuals possess a highly developed dorsolateral prefrontal cortex, which functions like a master air traffic controller. This region is responsible for code switching, the ability to flip between languages while actively inhibiting the vocabulary and grammar of the language not currently in use. Research published in the journal Nature indicates that learning a second language can actually increase the density of gray matter in the left inferior parietal cortex, physically reshaping the brain to accommodate new ways of expression. If you grew up speaking more than one language, do you find that your personality or even your sense of humor shifts slightly depending on which tongue you are using? This fluidity suggests that language is not merely a tool for description, but a framework that shapes our internal reality. The principle of linguistic relativity posits that the specific language we speak influences how we perceive the world around us. For example, speakers of languages that use cardinal directions instead of left and right possess an uncanny, internal compass that remains active even in windowless rooms. Similarly, the way a language categorizes colors can sharpen the brain's ability to distinguish between specific hues that speakers of other languages might see as identical. This indicates that the brain is not a passive observer of the world, but an active interpreter that uses the structure of language to filter and organize sensory data. Even the act of reading, a relatively recent human invention, requires the brain to hijack the visual word form area, a region originally evolved for recognizing objects and faces, and repurpose it for decoding symbols. This neuroplasticity allows us to bridge the gap between sight and sound, turning ink on a page into a vibrant internal monologue that carries the weight of human history. While we perceive the world as a steady flow, the brain is actually constructing our sense of time from a chaotic symphony of internal rhythms. It does not rely on a single central clock to measure the passing seconds. Instead, various neural circuits specialize in different scales of duration through a decentralized system. The cerebellum, tucked at the base of the skull, manages the split-second timing required for complex movement and fine motor coordination. Simultaneously, the basal ganglia deeper in the brain help us track intervals of seconds and minutes, allowing us to anticipate the beat of a song or the timing of a conversation. At the most fundamental biological level, the suprachiasmatic nucleus acts as a master pacemaker, synchronizing our internal chemistry with the 24-hour rotation of the planet. Have you ever noticed how a few seconds of intense fear can feel like an eternity? This distortion occurs because the amygdala, our emotional processing center, kicks into high gear during perceived threats. It commands the brain to record memories with a much higher resolution and density than usual. When you later reflect on a moment of danger, your mind sifts through a massive amount of detailed data, tricking your consciousness into believing the event lasted far longer than the physical clock suggests. Research by neuroscientist David Eagleman has shown that while we don't actually see in slow motion during a crisis, our recollection of the event expands because the file size of the memory is so much larger. This phenomenon of memory density also explains why time seems to accelerate as we grow older. To a child, every day is a landscape of novel experiences, requiring the brain to work intensely to process and store new sensory patterns. For an adult, the world becomes a series of predictable routines and familiar scripts. When the brain encounters less novelty, it encodes fewer distinct memory anchors, making weeks and months appear to blur together in retrospect. This is often referred to as the proportional theory, suggesting that as we age, each new year represents a smaller fraction of our total life experience, changing our subjective scale of duration. Our conscious experience is not a continuous stream but rather a series of discrete snapshots stitched together by the brain's internal refresh rate. Scientists measure this through flicker fusion frequency, the specific point at which a blinking light appears as a solid, steady beam. For the average human, this happens at roughly 60 cycles per second, though this rate can fluctuate based on our state of arousal. In moments of extreme focus, the brain can effectively increase its sampling rate, gathering more information per second to provide a competitive edge. Do you remember a time when you were so deeply immersed in a task that hours seemed to vanish in what felt like only 20 minutes? This state of flow demonstrates how the brain can prioritize task processing over time tracking, essentially silencing our internal metronome to maximize cognitive efficiency. This temporal flexibility proves that time is not just something we measure, but something we actively create. The brain manages an overwhelming torrent of data by deciding what deserves your consciousness and what should remain in the shadows. Consider the sensation of the fabric against your skin or the steady weight of your own body. Until this moment, your parietal lobe likely suppressed that information to prevent sensory overload. This process, known as sensory gating, allows the thalamus to act as a high-level filter, silencing the hum of a refrigerator or the rhythmic ticking of a distant engine. Without this neurological selective hearing, the world would be a chaotic cacophony of competing signals, making sleep or deep thought nearly impossible. This filtering is not just passive, it is an active, energy-intensive suppression of the irrelevant that allows the mind to maintain a stable internal environment. Our attention operates through two distinct pathways known as the bottom-up and top-down systems. Bottom-up attention is involuntary and primitive, triggered by sudden movements, bright flashes, or sharp sounds that demand immediate processing. It serves as an ancient survival mechanism, ensuring that we react to potential threats before we even consciously perceive them. In contrast, top-down attention is a sophisticated superpower of the prefrontal cortex that allows you to willfully direct your focus toward a single page of a book or a specific voice in a crowded room. This intentional steering of cognitive resources is what enables humans to ignore immediate distractions and pursue complex, long-term goals. Do you find it easier to concentrate on tasks when the room is completely silent, Or do you prefer a low, steady background hum? When we achieve a state of deep immersion, the brain's chemistry undergoes a fascinating shift to sustain that focus over long durations. During these flow states, the prefrontal cortex experiences transient hypofrontality, where certain executive areas temporarily quiet down to allow for faster, more fluid processing. A precise cocktail of dopamine, endorphins, and norepinephrine floods the neural pathways, heightening pattern recognition while dampening the sense of self-consciousness. This neurochemical reward system turns intense mental effort into a self-sustaining loop, where the challenge of the task perfectly matches your internal skill level. However, this delicate balance is easily disrupted by the modern habit of multitasking, which is actually a process of rapid, exhausting context switching. Every time the brain shifts focus between different streams of information, it incurs a switching cost that depletes vital metabolic resources. Each transition requires the brain to burn through significant amounts of oxygenated glucose, leading to mental fatigue much faster than sustained focus would. Research published in the journal Nature indicates that these micro-interruptions can lower effective cognitive performance during the task and increase the production of cortisol, the body's primary stress hormone. By understanding that focus is a finite biological resource, we can appreciate why protecting our attention is essential for cognitive longevity. The brain is not a parallel processor for high-level tasks. It is a serial one that performs best when it can commit its full metabolic wealth to a single, unfolding thought. Deep within the temporal lobe, there sits an almond-shaped cluster of neurons that serves as the brain's primary alarm system. This structure, the amygdala, is tirelessly vigilant, monitoring the environment for anything that might signal a threat to our safety. It is essentially a master of pattern recognition for danger, working at speeds that far outpace our conscious awareness. When the eyes perceive a shape in the grass that resembles a snake, the amygdala initiates a response before the visual cortex has even finished identifying the object. This rapid processing is a remnant of our evolutionary history, a hidden superpower that prioritizes immediate survival over precise accuracy. In moments of intense stress, we experience what is known as an amygdala hijack, where this emotional center effectively seizes control of the neural circuitry. It sends urgent signals to the hypothalamus, which then activates the sympathetic nervous system, triggering the well-known fight-or-flight response. During this event, the prefrontal cortex, the part of the brain that handles logic, planning, and social nuance, is temporarily sidelined. This ensures that the body's metabolic resources are diverted toward physical action rather than abstract thought. It is a biological emergency protocol that operates with a singular focus, ignoring the complexities of the modern world in favor of ancient survival tactics. The amygdala is also the architect of fear conditioning, the process by which we learn to avoid specific dangers through experience. This involves the creation of powerful associative memories that link sensory inputs to emotional reactions. When a negative event occurs, the amygdala works in tandem with the hippocampus to ensure the context of that event is never forgotten. This is how the brain creates a map of the world's hazards, though sometimes the map becomes overly sensitive. Phobias are essentially high-definition fear memories that the amygdala refuses to dampen, even when the original threat is long gone. Interestingly, Recent neuroscience research suggests that much of what we perceive as anxiety is actually a byproduct of prediction errors within these deep brain structures. The brain is constantly trying to guess what will happen next, and when there is a mismatch between its expectations and reality, the amygdala may signal an alarm. For those with high levels of anxiety, the amygdala's threshold for these prediction errors is often lower, leading the brain to overcalculate risk. This reveals a profound truth about our biology. Our brains are not designed to keep us happy, but to keep us alive. Do you find that your mind tends to revisit past mistakes more often during the quiet hours of the evening? This cycle of internal monitoring is the brain's way of trying to refine its predictive models, ensuring that the same mistake is not repeated. By viewing these intense emotional responses through the lens of neurobiology, we can appreciate the sophisticated, albeit sometimes overzealous, protective mechanisms that have allowed our species to endure for millennia. These circuits ensure that even in sleep, the brain remains a dedicated guardian of our existence. While we often associate productivity with intense focus, the brain possesses an equally powerful mode that activates only when we let go of specific tasks. This state involves a specialized circuit known as the default mode network, or DMN. It becomes most active during moments of daydreaming, mind-wandering, or simple boredom. Far from being a waste of metabolic energy, the DMN is a highly sophisticated processing engine that integrates past experiences with future possibilities. When the prefrontal cortex relaxes its grip on logical, linear thinking, the DMN begins to scan the vast archives of the subconscious. It bridges distant regions of the cortex that rarely communicate during the workday. This is why solutions to complex problems often surface when you are performing a mundane task like washing dishes or walking through a park. The brain is utilizing a form of background processing that operates outside of conscious awareness. Neuroscientists have observed that during these periods of relaxation, the brain's filter becomes more permeable. It allows for the collision of seemingly unrelated concepts, a process fundamental to original thought. This state is sometimes referred to as transient hypofrontality, where the executive centers of the brain momentarily step back, allowing more spontaneous and divergent thoughts to emerge. Think of the brain as a vast library where the lights are dimmed. Suddenly, you might notice how a book on architecture shares a structural rhythm with a volume on music theory. Have you ever noticed how your best ideas seem to arrive when you aren't actually looking for them? This phenomenon occurs because the brain is less restricted by the need for immediate, practical results. The incubation period of an idea is a real biological phase where the brain works silently to weave together disparate threads of information. The right hemisphere plays a critical role in this sudden leap of intuition. While the left hemisphere is often specialized for language and granular detail, the right hemisphere excels at detecting broad patterns and metaphorical connections. Research involving EEG and fMRI scans shows a sudden burst of high-frequency gamma waves in the right superior temporal gyrus just moments before a person experiences a flash of insight. This aha moment represents the brain successfully synthesizing a new pattern from fragmented data. It is a biological celebration of a connection finally being made. These insights are not random. They are the result of the brain's relentless drive to find order and meaning in the chaos of sensory input. At its biological core, creativity is not the act of creating something out of nothing. Instead, It is the sophisticated recombination of existing memory fragments. Every image, sound, and fact stored in the hippocampus and neocortex serves as a building block. The creative superpower lies in the brain's ability to dismantle these memories and reassemble them into entirely new configurations. This explains why diverse experiences are so vital for innovation. The more varied the internal library, the more materials the brain has for its architectural experiments. The neural pathways for memory and imagination are remarkably similar, suggesting that the brain uses the same machinery to remember the past as it does to invent the future. By drawing on this inner reservoir, the brain transforms old information into fresh perspectives, ensuring that our cognitive landscape is always evolving. This internal evolution is driven by a complex chemical alphabet known as neurotransmitters. These molecules act as the essential bridge between electrical impulses and biological action, translating a silent spark of energy into a feeling, a movement, or a memory. Every thought you experience is accompanied by a microscopic spray of these compounds across synaptic gaps, the tiny spaces between neurons. This chemical signaling is so precise that a single neuron can receive thousands of different inputs simultaneously, yet it distinguishes between them with incredible accuracy. This fluid language allows the brain to communicate with itself and the rest of the body in real time. Among the most influential of these messengers is dopamine, which is frequently mischaracterized as the simple pursuit of pleasure. In reality, neuroscientists at Harvard University describe dopamine as the chemical of anticipation and motivation. It surges not when we receive a reward, but when we perceive the possibility of one. This system keeps the brain focused on the future, driving the persistence needed to learn a new skill or navigate a difficult task. When dopamine levels are balanced, we feel a sense of purposeful drive and mental clarity. Do you find that you are more productive when you have a specific goal in mind, or do you prefer to let your curiosity lead the way? This drive is what allows us to endure temporary discomfort for the sake of a long-term achievement. While dopamine pushes us toward the new, serotonin helps us find stability in the present. This molecule is a primary regulator of mood, appetite, and the sleep-wake cycle. It acts as a biological stabilizer, smoothing out the peaks and valleys of our emotional landscape. Interestingly, the vast majority of the body's serotonin is produced in the gastrointestinal tract, highlighting a direct chemical link between the gut and the brain's emotional centers. High levels of serotonin are associated with feelings of safety and social confidence, while lower levels can contribute to a sense of unease. By maintaining this chemical equilibrium, the brain ensures that we remain resilient in the face of environmental changes. Human connection is further facilitated by oxytocin, a neuropeptide that functions as a powerful social glue. Often referred to as the bonding hormone, oxytocin is released during moments of physical touch, eye contact, and even during deep, meaningful conversations. It works by dampening the activity of the amygdala, the region responsible for processing fear and social anxiety. By reducing our natural wariness, oxytocin makes it possible to form deep bonds of trust and cooperation with others. This chemical foundation is what allows for the creation of families, friendships, and large-scale communities. It effectively transforms a group of strangers into a cohesive social unit. When the environment presents a challenge, the brain shifts its chemistry toward survival. The stress response system triggers the release of adrenaline, also known as epinephrine, which instantly redirects the body's resources. Heart rate increases, air passages dilate, and energy is diverted away from non-essential functions like digestion to fuel the muscles and brain. This is followed by a release of cortisol, which helps maintain high energy levels during a prolonged period of pressure. While these chemicals are vital for handling immediate threats, the brain is designed to return to a state of calm once the challenge has passed. This rhythmic fluctuation between high alert states and restorative periods is essential for maintaining cognitive health. Every molecule works in concert, ensuring that the brain remains a highly responsive and adaptive organ, capable of managing both the quietest reflections and the most intense physical demands. Neurons are essentially high-performance engines that require an unrelenting stream of fuel to maintain their structural integrity and electrical signaling. While the human brain accounts for only about 2% of total body weight, it greedily consumes roughly 20% of the oxygen and calories provided by the cardiovascular system. This constant influx of gas is non-negotiable because, unlike muscle cells, neurons possess almost no internal energy stores, making them uniquely vulnerable to any interruption in flow. Within just a few minutes of oxygen deprivation, the delicate electrochemical gradients that allow cells to communicate begin to collapse in a process known as the ischemic cascade. It is a sobering reality of biology that while other tissues can shift into anaerobic metabolism to survive temporary shortages, the brain relies almost exclusively on aerobic respiration to keep its intricate architecture from unraveling. Protecting this fragile neural landscape is a highly selective security perimeter known as the blood-brain barrier. This isn't a physical wall in the traditional sense, but rather a sophisticated physiological gatekeeper formed by tightly packed endothelial cells lining the cerebral capillaries. These cells create junctions so narrow that most bacteria and large, potentially harmful molecules are physically blocked from entering the neural environment. However, the barrier is intelligently permeable, utilizing specialized transport proteins to usher in essential nutrients like glucose while simultaneously flushing out metabolic waste products. Do you ever find yourself wondering how your mind maintains its clarity even when the rest of your body is under stress? Whenever a specific region of the cortex becomes more active, the brain initiates a localized increase in blood flow to that exact coordinate. This phenomenon, called neurovascular coupling, serves as the fundamental principle behind functional magnetic resonance imaging, which allows scientists to see the brain in action. Astrocytes, the star-shaped glial cells, act as the primary intermediaries that sense neuronal firing and signal the surrounding blood vessels to dilate. This ensures that the hardest-working neurons receive a surplus of oxygenated hemoglobin, which actually overcompensates for the immediate demand. Such rapid vascular responsiveness occasionally leads to the sharp, fleeting sensation of a brain freeze. Formerly termed sphenopalatine ganglioneuralgia, this happens when something cold touches the roof of the mouth, causing a rapid constriction and subsequent massive dilation of the internal carotid artery. The brain perceives this sudden rush of blood as a distress signal, transmitting a referred pain through the trigeminal nerve directly to the forehead. It serves as a fascinating example of how the brain's protective vascular reflexes can sometimes result in temporary discomfort while attempting to maintain thermal stability. Even during deep rest, these vessels continue to pulse with a rhythmic elegance, clearing away the day's molecular debris through the glymphatic system. This internal maintenance is a silent testament to the biological cost of our most distinctive feature, the sheer complexity of the human cerebral cortex. While primates like chimpanzees and bonobos share much of our genetic code, the human brain has expanded in ways that prioritize cognitive processing over raw physical power. In the human cortex, there are approximately 16 billion neurons dedicated specifically to high-level functions, which is more than double the number found in a gorilla, despite the gorilla's much larger body size. This dense packing of cells is facilitated by an intense degree of gyrification or folding, which allows a massive surface area to fit within the constraints of the skull. This folding isn't random. It follows a precise blueprint that maximizes the speed of communication between distant neural hubs. One of the most profound leaps in our evolutionary journey is the disproportionate growth of the prefrontal cortex. This region, located just behind the forehead, serves as the brain's executive suite, responsible for what scientists call mental time travel. While most animals are tethered to the present moment, reacting to immediate stimuli, the human prefrontal cortex allows us to simulate the future. We can weigh the consequences of actions that have not yet occurred or reflect on the distant past to refine our current behavior. This capacity for abstraction transformed our ancestors from reactive foragers into strategic planners. To support this specialized hardware, the human body had to make a significant metabolic sacrifice. The brain is an exceptionally hungry organ, consuming roughly 20% of the body's total energy despite making up only 2% of its weight. To afford this high-wattage intelligence, our species underwent what researchers call the expensive tissue hypothesis. This theory suggests that humans traded the energy-intensive digestive systems and massive muscle groups seen in other primates for a more efficient, calorie-dense diet and a larger brain. As a result, a human is significantly weaker than a chimpanzee of the same weight, as our biological resources are diverted away from muscle fibers and toward neural pathways. This trade-off proved to be an incredible advantage, as it gave rise to the ability to manipulate the world through sophisticated tool use. The brain possesses a remarkable trait called distal attribution, which allows it to incorporate external objects into its own internal map of the body. When you pick up a tool, the neurons in your parietal cortex begin to fire as if the object is a literal extension of your limb. This fluid boundary between the self and the environment transformed the way we interact with the physical world, allowing the mind to project its intent through wood, stone, and eventually digital interfaces. This neural flexibility ensures that our capabilities are not limited by our biological reach, but are instead expanded by every instrument we master. It is this unique combination of foresight, energy redirection, and environmental integration that defines the hidden power of the human mind. The sheer capacity for rewiring suggests that the brain is less like a hardwired computer and more like a living, breathing ecosystem that constantly reclaims lost territory. When one sense is lost, the underlying neural real estate is rarely left vacant. Instead, the cortex begins to reallocate resources to process information from the remaining senses, often turning a blind individual's visual cortex into a sophisticated center for touch and sound. This resilience is what allows us to recover from physical trauma and learn entirely new languages even into late adulthood, proving that our cognitive map is never truly finished or fixed. It is a masterpiece of biological efficiency, managed by roughly 86 billion neurons that consume less energy than a dim light bulb, yet outperform the most advanced supercomputers in pattern recognition and creative synthesis. Despite this mechanical understanding of synapses and electrical impulses, we eventually arrive at the most profound mystery in all of science, often called the hard problem of consciousness. While we can map the neurobiology of a sunset, tracking how photons hit the retina and trigger electrical signals in the primary visual cortex, we still cannot explain how those physical processes transform into the subjective, private feeling of seeing the color gold. Philosophers and neuroscientists like David Chalmers struggle to bridge the gap between physical gray matter and the vivid internal experience of being alive. There is no specific consciousness neuron or a single location in the brain where the self resides. Instead, our sense of identity seems to emerge from the complex, high-speed choreography of the entire network, a biological symphony that remains largely unwritten and misunderstood. The frontiers of neuroscience are currently expanding into the vast majority of brain cells that are not neurons, known as glia. For over a century, these were dismissed as mere glue or support structures, but recent research indicates they may play a critical role in information processing, potentially communicating through subtle calcium waves that operate on a different timescale than neurons. We are also just starting to uncover the mysteries of the glymphatic system, the brain's unique waste clearance mechanism that operates primarily while we sleep, washing away metabolic debris that accumulates during our waking hours. How do these microscopic currents influence our long-term health and the preservation of our personality over a lifetime? Much of the brain remains a terra incognita, a vast landscape where unknown chemical dialogues occur every millisecond, hidden from our direct awareness. Consider the sheer complexity of what is happening as you process these words in this moment of quiet. Your brain is decoding symbols, turning them into internal sound, and then into meaning, all while maintaining your heartbeat and regulating your temperature without a single conscious thought. Do you ever wonder how many of your daily decisions are truly yours, rather than the result of these deep, automated systems working beneath the surface? As you settle into the stillness, know that your mind is still working, weaving together the day's events into the permanent tapestry of your memory. This internal universe is perhaps the greatest wonder we will ever encounter. If you enjoy uncovering the quiet truths of our world, please consider subscribing to join us for more journeys through the wonders of science. The human brain is a testament to the fact that even in our most quiet moments, we are carrying a power that is truly limitless.